so our topic today will be transport in cells. So let's begin with our goal. By the end of this video, you should be able to explain the two ways that materials can move in and out of cells. So let's begin with this question. Why do materials need to move in and out of the cell? Why is this so important? And the simple reason is that the cell has to stay alive, and before it can do that, it needs some things to use. Example, what? We have water or oxygen or glucose, which the cell will use for energy to stay alive. And also the cell will produce some toxic materials which it has to remove into the outside environment. So things has to move in and out of the cell to keep the cell alive, to give the cell the materials it needs to survive. And all of these processes help to balance the inside environment with the outside environment of the cell. We call homeostasis. Now, this process is regulated by the cell membrane. So it's very important to look at the different parts of the cell membrane and the structure of the cell membrane. So quickly, this is the phospholipid bilayer. It's a cell membrane. It's a phospholipid bilayer. And if you look carefully in this picture, we have some cholesterol molecules inside the phospholipid, which will help to hold them together. We also have some proteins inside, and their job is to help to move materials. There are two types here. We have the channel proteins and the carrier proteins. The difference between them is that the carrier proteins can open and close, but the channel proteins stay open all the time. And we also have glycoproteins, which are just glucose joined together with some proteins. And we have glycolipids, which is glucose joined with the lipid. And they all have very important purposes for the cell. Now, the cell membrane is termed as semi-permeable membrane because it can choose materials to pass and some materials not to pass. Now, let's look at the two ways that materials move in and out of the cell. The first one is termed as passive transport. And in passive transport, the cell does not use energy. Example is when the cell is trying to transport water or oxygen or CO2, small molecules. Number two is active transport, in which the cell actually needs energy to move the materials in or out. Maybe because the materials are very large, like proteins. So let's take a look at passive transport and active transport, starting with the first example of passive transport called simple diffusion. Now, in this picture right here is a colorful experiment about a cabbage inside a dye solution. And what you can see here is that the colored dye or the dye molecules are moving into the cabbage cells, making them very colorful. Now, if we zoom into the cells of the cabbage, what are we going to see? And this is what we may see, or we will see, is that outside the cabbage cells, we have a high concentration of the dye molecules. But inside the cabbage cell, we have low concentration. So what happens is that the molecules, the dye molecules, will move from high concentration area to low concentration area. And this is called simple diffusion. Now, let's take a look at these three situations. In A, we have 3 molecules of oxygen outside and 1 inside the cell. We have in B, 1, 2, 3, 4, 5, 6, 7. 7 molecules of oxygen outside, 2 molecules inside the cell. And see, we have equal amount of oxygen molecules inside and outside. I want you to consider this question. In which situation will the rate of simple diffusion be highest? Is it A, B, or C? Now, if you're thinking the answer is B, you are right. This is because of something we call concentration gradient. And that is what? The difference between the concentration of the two areas. So we can see that in B, we have the highest difference in concentration. So the higher the difference in concentration, the faster the diffusion. Okay. So let's look at our second example of passive transport, and that is facilitated diffusion. In facilitated diffusion, the molecules are still moving from high concentration area to low concentration area. But the difference is we need proteins to help to move the materials. Maybe because the materials are a bit large, like glucose, or they simply cannot pass the phospholipid bilayer. Okay, so this is facilitated diffusion. There is no use of energy, but we need proteins to help to move the materials. So the first example of active transport is the sodium-potassium pump, which is very important for our cells because they help to move sodium ions out of the cells and bring potassium ions inside the cell. Now, how does this happen? Okay, if you look outside the cell, you can see we have high concentration of sodium, the pink round object. Inside we have low concentration. So we're trying to move the sodium from inside from a low concentration to a high concentration, which is opposite of passive transport. We go in against the concentration gradient. So we need energy, and we get the energy from ATP. So let's watch here. We have 3 ions of sodium going to the protein channel, and the energy from ATP will help to open the protein channel. As it opens, we have 2 potassium ions going inside the channel protein or the carrier protein, and finally to move inside. Again, the energy is supplied by ATP as 3 sodium ions move from inside to outside the cell against the concentration gradient. We have 2 potassium ions moving into the cell, also against the concentration gradient. This is what the sodium-potassium pump. The second type of active transport is called endocytosis, and endo means in, cyto means cell. So endocytosis is when we move large particles inside the cell. Example is when you try to transport a solid material into the cell, which we term as cell eating or phagocytosis. A second type of endocytosis when we try to transport liquid materials inside the cell, which is termed as cell drinking or pinocytosis. And finally, we have the receptor-mediated endocytosis, and that is what? The materials will move by help of the receptors outside the cell membrane. And this is how viruses enter our cells. Lastly, we have exocytosis. You can see in this picture, materials will move from inside the cell to outside the cell. Exo means out, cyto means cell, and the exocytosis is what? When materials move from inside to outside the cell. So thank you very much for watching. I hope this is very helpful. See you in the next video.